Okay, so welcome to this week's online lab, which is focusing on Streptococcus. I actually consider this a continuation of last week's lab because Staphylococcus and Streptococcus go together very well, and you have to basically be able to kind of ask yourself when encountering an infection of either of these types, well, how do I distinguish them? How do I first distinguish between Staph and Strep? How do I then figure out exactly which species within either of these genuses is currently the problem? Now, the quiz next week, which will be open from Sunday through Wednesday, will be on both Staph and Strep. So on the previous online lab as well as this one. Now, just a quick reminder from the previous lab, some important points that you have to remember for both of these genuses. First of all, we said both Staph and Strep are two of the most important genuses because they cause a lot of the major infections, especially for any of you going into the medical field who will be working in hospitals. Huge, huge cause of many of the hospital infections. Now, it's also important to kind of think of the similarities and differences. As we mentioned, both of these Staph and Strep are gram-positive and coccus shaped, which means that if you gram stain them, what color will they be? What shape will they be? Well, they'll be purple and they will be round. But the way that they'll differ in that gram stain is that Staphylococci will be in clusters like grapes, whereas Streptococci will be in the strips. Remember, strep, strip, or strip of streps. Either way, they're linear, chain-like arrangement. The other thing I want you to remember from the previous lab and to put circles and stars and highlight is how would you distinguish between Staph and Strep. So for instance, I could ask you if a patient is suffering from an infection and you're not sure whether it's Staph or it's Strep, what's a quick test that you would be able to do on their culture to distinguish between the two to help you then narrow down the problem? The answer would be the catalase test. You should also be able to explain, what the premise is, why you see the bubbling in one of these images, but you do not see it in the other. So the idea, again, with the catalase test is that Staphylococcus is catalase positive. They produce the enzyme catalase that when that bacteria encounters hydrogen peroxide, they're able to break it down. One of the products of it being broken down is oxygen. So you end up seeing anytime that bacteria comes in contact with hydrogen peroxide, you see that bubbling effect. Again, the way you could remember this or think of it is when you have a cut. If anyone has ever put hydrogen peroxide on a cut, you see that bubbling because your skin is so chock full of Staphylococcus. Okay? Whereas Streptococcus, you notice in the slide, there is no bubbling because they do not produce the enzyme. Okay, make sure you're very comfortable explaining that. So now enough about the reminders and the review. Let's get to Streptococcus. Now, out of the two that we've been talking about, shockingly enough, Streptococcus is responsible for more infectious diseases than most other genuses, a lot of times even more than Staph. Now, when you encounter Streptococcus, there are two main ways that they will categorize or classify the different species within this genus. The two major methods of categorizing them are either by the species hemolytic activity or their serological classification. And I want you to be very comfortable with explaining what each of these terms means. So hemolytic we encountered last time. When you hear hemolytic or hemolysis, this means that the bacteria is capable of breaking down your red blood cells. Remember we said that there are three types of hemolytic activity that you have to know. There's alpha, beta, and gamma. But we usually organize them in the order of beta, alpha, and gamma, going from the most pathogenic to the least pathogenic. So remember the trick we said, beta, you can remember that's the worst by saying beta bad. Beta hemolysis or beta hemolytic activity is the most pathogenic because that is the complete breakdown of those red blood cells, okay? You kind of need your red blood cells, so that's a problem. The second one is alpha hemolytic activity. Alpha hemolytic activity is only the partial breakdown of the red blood cells. And I know I say only the partial breakdown as if it's not that bad, when it's still a problem. You know, you don't want any breakdown of your red blood cells, but it's not as bad as the complete breakdown. The third one is then gamma, and gamma means little to no breakdown of the red blood cells, okay? So that one's not very pathogenic. So you'd like your microbes to be gamma hemolytic so that they're not causing you much harm. The next way of categorizing or classifying Streptococcus species is by serological means. This is something that's going to come back in lecture and lab multiple times, that term serological or serology. What that means is you are looking at antibodies and antigens, okay? Remember, antibodies are what your body produces to protect you, and antigens are those little surface proteins on any microbes or cells. They're basically the name tags, okay? Antibodies then bind to antigens, okay? We'll go more into that in the immune system lesson that we do. But now, when we talk about serological classification, that's basically meaning that we can classify these different streptococci species based on which antigens or which proteins they have on their cell surface, okay? We end up breaking them down into about 20 groups, with A, B, C, and D being the ones that are mainly human pathogens. Okay. Notice that I point out that D group is actually no longer called group D. It's actually called Enterococcus. Now, in this online lab, the three that we're going to focus on are group A, B, and Enterococcus, because these are three of the ones that have some species that cause a lot of human infections. Okay, so let's meet our group A, B, and D. Now keep in mind, with each of these groups, they contain a whole bunch of different species, but the three species that we are going to focus on in this lab and distinguish amongst are Strep pyogenes, Strep agalactiae, and E. faecalis. Now, I know you're probably not listening to a word I'm saying right now because you're too busy vomiting somewhere after looking at the lovely pictures that I placed on this slide. But I basically wanted to give you kind of the take-home message pretty visually and obviously of which of the three is going to be the worst. So today we're going to be identifying group A, Strep pyogenes, group B, agalactiae, and D, which is now Enterococcus, E. faecalis. But out of the three, the ones that these lovely pictures are actually Strep pyogenes cause these. Now, what's interesting about Strep pyogenes is you've all probably heard about this strep before, and many of you have actually probably had it. Now, not to the extent that you're seeing in these pictures, but Strep pyogenes is an interesting microbe because it is the cause of the common sore throat, strep throat. So this microbe can cause something as mild as strep throat or sore throat, but at something as severe as necrotizing fasciitis. So it's kind of scary when you think of. Now, Strep pyogenes we're going to go over in lecture. There's a whole range of things that it can cause. In addition to sore throat, it can cause pharyngitis. In addition to that throat region, it can also extend to causing scarlet fever, so depending on whether or not it's producing toxins. And as you can see in the pictures here, it can also cause necrotizing fasciitis or forms of toxic shock syndrome, which we also saw in Staph as well. So Strep pyogenes is the big bad guy of today's lesson, because as you can see, it can be pretty damaging depending on where it's located, whether or not it's made it into your bloodstream, so if it's gone septic, and whether or not it's producing toxins, which, as you know, toxins, especially exotoxins, can be very damaging. The second group that we have here is group B, and the species we're going to focus on for that is Strep agalactiae. Now, Strep agalactiae is mainly known for being present in vaginal mucosa and can cause neonatal infections because of this location. You then have E. faecalis, which again is not usually as severe as Strep pyogenes. This one you usually find causing infections in the lung or in the UTI, as a UTI, or if it enters the bloodstream, sometimes it causes problems, and usually the way it enters the bloodstream is from intestinal lacerations, so especially being careful in terms of surgeries or issues like that. But again, out of the three, the one that's the worst that you're going to want to be able to distinguish is Strep pyogenes. Now, there are a few ways to distinguish amongst those three, okay? That's going to be the goal of today's lab, is finding different ways to kind of identify, does your patient have pyogenes, agalactiae, or E. faecalis? Now, the first test that we can do here is hemolysis, or hemolytic activity. Now, you can see on this slide the difference amongst the strains. So Strep pyogenes and agalactiae, they're both beta hemolytic, whereas E. faecalis is alpha to gamma hemolytic. So you have to ask yourself, if I'm testing or distinguishing based on hemolytic activity, how would I do that? How would I identify which is which? Well, hemolytic activity, we already mentioned, means the breakdown of red blood cells. So the way that you would test for this is by using blood agar plates. These blood agar plates usually have 5% sheep blood in them, which is what gives them that reddish kind of color. You then take a look where you streak your bacteria, and once the bacteria grows, you see how does it affect the red blood cells in that plate. So this first plate here shows beta hemolytic growth, where it looks almost like it's bright yellow because of the fact that these bacteria, they completely break down the red blood cells. So they completely remove the red color wherever the bacteria is, which makes that plate end up being transparent now. Instead of being solid red, it's now transparent. So the light shines through it. So that gives that appearance of the see-through, almost glowing look to it. The middle one, instead now it's turned that greenish kind of brownish color because that's alpha hemolytic activity, where the bacteria started to break down the red blood cells a little bit, but didn't fully break them down. So you can't completely see through that plate now. The last one is gamma, where there's no breakdown and the plate looks just as solid red as before. So you won't see the light really shining through. Okay, so what you would do is you streak the bacteria on the blood agar plate and you look to see whether or not the bacteria chewed through the red blood cells. Is that plate, is the growth now looking clear and bright light coming through? Or did the plate stay fairly the same as before and only kind of change slightly to like a greenish brown color? So pyogenes and agalactiae, because they're beta hemolytic, they would look like this plate. Clear, see-through, almost glowing yellow, right? E. faecalis on a blood agar plate would instead look like either of these two, okay? Because depending on the exact strain you're dealing with of E. faecalis, there'll be little to no hemolytic activity, okay? So make sure you're comfortable with understanding what hemolytic activity is, what test you would do for it, how you would set up that test, and what the results would tell you. Another way to test for, which of these three strains you're dealing with is to use Bacitracin, which is an antibiotic, and you've probably heard of it or seen the name, especially in things like triple antibiotic. Bacitracin is one of the ones in there. Now, before we get to this one, I want to remind you, For staph strains, if you're dealing with Staphylococcus, which antibiotic do you want to use to distinguish? For Staphylococcus, it's novobiacin, okay, novobiacin, whereas for Streptococcus, you use Bacitracin. Now, there's a trick to remember which of the 3 strep that we're dealing with today is sensitive to Bacitracin. The other 2 are not. The way to remember which one is sensitive is to say Bacitracin eats pie, okay? If anyone likes pie, whether it's apple pie, or banana pie, whatever it is, you remember based on that little trick, because Bacitracin will kill or chew up on the plate, as you see here, pyogenes, strep pyogenes, okay? So if you remember Bacitracin eats pie, then whenever you see a plate with a Bacitracin disc and you see a circle around it, a zone of inhibition, where that disc has killed off or eaten up all of the bacteria around it, that tells you this bacteria here is pyogenes. The bacteria streaked where you do not see a zone of inhibition, the Bacitracin has not killed off any of the bacteria, that would tell you that on this side of the plate is either Agalactiae or E. faecalis. Both of them are resistant to Bacitracin. Okay, so if I ever asked you of how you would want to identify if the patient has strep pyogenes compared to the other 2 that we're dealing with today, one of the quick ways to kind of distinguish pyogenes from the others is to do the Bacitracin test. So you would tell me you would streak bacteria on a plate, place a disc containing Bacitracin in the center of where you streak that bacteria, and if you see a zone of inhibition, a big circle around that disc, you know that there's pyogenes there, whereas E. faecalis and Agalactiae will not have that zone of inhibition because they're resistant. The next one that we have here is distinguishing with Bile Esculin. And I love the Bile Esculin test because this is the one that from across the room in our giant lab, you can identify E. faecalis. Okay, so E. faecalis is the strain that you will identify using the Bile Esculin test. What happens is you take this tube of Bile Esculin agar. So it looks liquidy, but that's actually a solid slant of agar that starts out the regular yellowish beige color. And what you do is you swab the bacteria on the slanted portion. If that agar turns black, that tells you that E. faecalis is present because E. faecalis hydrolyzes the esculin that's in that media. And the products from it hydrolyzing the esculin basically interact with that media, and it causes these insoluble iron salts. And if you've ever played with those games that they've had where you take the magnet and you're moving the little pieces around the board, you've seen the color is that dark blackish brown, okay, of iron salts. And so whenever you see the Bile Esculin test turn that dark color, you know E. faecalis is present, and the reason it changed that color is because E. faecalis is hydrolyzing the esculin. Now there's a trick to remember which of the strains the Bile Esculin test is showing you, and that changed to blackish brown. And the trick that I always use is think of the name E. faecalis. When you hear faecalis, that sounds like fecal. Fecal as in poop. So when you see the Bile Esculin test that the tube turned a dark blackish brownish color, it turned a poop color, that is E. faecalis, okay? So make sure you're very comfortable with knowing the different tests that you can quickly identify a particular strain out of the 3 species that we're talking about. So for instance, Bacitracin showed us pyogenes. Bile Esculin quickly shows us E. faecalis, and then the one that's left over out of that, you know is Agalactiae. Now, it's always good to have a little extra testing, kind of think of it as a second opinion or to just kind of prove that what you're seeing is true. So you can also look at halotolerance, okay? Pyogenes and Agalactiae are not halotolerant, whereas E. faecalis is halotolerant. Now, what do you think that means when you see halotolerant? Well, it sounds kind of like halophile, which you're all pros and experts at now. So halotolerant means that E. faecalis is the one that will be able to withstand a little higher salt content than the other two. Pyogenes and Agalactiae will not grow in the presence of, 3% salt, for instance. So what you can do to test this, well, you've all worked with salt plates already, or even if you have a selective plate like mannitol salt agar, so MSA plates. If you streak the 3 strains on MSA plates, Pyogenes and Agalactiae usually will not grow, whereas E. faecalis will grow because it can tolerate the salt in the MSA plate. Okay, so it's always good to have an extra little way to double check your results. So now we get to the fun part, interpreting 3 sets of results. Now I'm going to just give you these slides and I'm not going to tell you what the tests are or, what you're basically looking at. Based on listening to the narration and seeing the previous slides, you should be able to look at a tube or a plate with a result and be able to tell me, what test is that? Is it Bile Esculin? Is it Bacitracin? Are you looking at hemolytic activity? Okay, you should be able to tell me that type of information. You should be able to tell me why are you seeing the changes you see. Okay, so for instance, if there's a change in color, if there is a change in the plate's transparency, if there's a circle, a zone of inhibition or not, you should be able to explain why these things are occurring, and you should be able to identify. And notice that sometimes you can't identify a single strain. Sometimes you're just narrowing down. So it's okay for you to tell me that based on this test, okay, for instance, Bile Esculin, if it doesn't turn black or brown, well, that tells you that it's not E. faecalis. So you've narrowed it down to either Agalactiae and Pyogenes, and you should then be able to tell me the next step that you would do. Okay, so don't be worried if you look at a result and you're like, oh, wait, I can't tell the exact species, I could just narrow it down. Sometimes that's results, okay? So contact me if you have any questions while you're going through these results. Okay, so here is the first set of results that you will be asked to analyze and interpret. And notice that I put the bacterial identities on them in terms of unknown identities. So this will be tube D, tube E, tube F. You then have set number 2. So you're looking at bacteria F and bacteria E. Now notice the plate has a line drawn down the center. That's so that you can see this is where the bacteria was streaked. This is how the plate looked uninoculated, so before any bacteria was placed. Okay, so this is the regular plate. This is what happens to it with bacteria F. This is the regular plate. This is what it looks like with bacteria E. And then we get to the final set, set number 3. And for set number 3, you're dealing with 2 of the bacteria again. Now it is D and F. Both of these plates represent F. It's just showing you from the front side versus the back side. Okay, and so here I drew the circle for you to see that circle outline, okay, versus bacteria D. Okay, so now you'll be able to do the post-lab where you will be asked to interpret these results and kind of tell us as much as you can about what the test is, what you're identifying, what valuable information it gave. Okay, as always, let me know if you have any questions or concerns through the Remind app, and have a great day.